On January 5th, 2010, at the Topsham Public Library, the Cadance River Education Alliance was proud to support Mount Ararat students from Glen Evans' sophomore biology class as they joined together to present their work. This poster symposium represented many days in the field by Glen Evans' class as they studied various issues pertaining to biology and their local environment. The place where this was conducted was the nearby 235-acre Cadance Preserve and Ecology Center. Their work, in many ways, represents CREA's 10-year journey. The story of the Cadance River Education Alliance is a story of compromise and is a great story between environmental and developer interests. Attending this event were several people who together back in 2000 formed a group called Topsham's Future that helped ultimately create the preserve and helped start CREA. Dana Carey, as well as Carla and John Rensenbrink, were in attendance and were amazed at how far this organization has come.
The other part of the course tonight, all of the displays, all of the exhibits, all of the things that the students have done in each of these ways of doing it fulfills very strongly the reason why John Wozleski and I in April of 2000 felt that this is what we wanted to have started, which is to really get the schools in the area, students, high school students, pupils in the elementary schools, and their teachers, and their superintendents, if we could, to get them really aroused about ecological education.
We did the large mammal study. We planted 2 cameras, one off the trail on a tributary in winter ways, and that's this area right here. And the other one was located right next to the river on a muddy bank where we found lots of beaver tracks, and we got the picture of the muskrat there. It's sort of hard to see, but you can see its tail.
And we just checked every week, and we put them on our computer. We found that in this area there were a lot of deer that we saw a lot of beaver tracks with, and
along with a porcupine, which we got a video of it crawling over our camera, which is pretty cool.
So we were studying reindeer lichen, and there's hundreds of different kinds of lichen. There's some that sticks onto rocks, and there's others that, like reindeer lichen, grow straight up from the soil. Reindeer lichen have very shallow roots. They're very small, and they can grow in less than about 2 centimeters of soil.
Reindeer lichen generally grow in tundra area, or it can grow in that area. The reason it has its name reindeer lichen is because our reindeer, caribou, and other that sort of feed off it as one of the main food source, especially in the winter time. Reindeer lichen don't generally need a lot of moisture, but it will use that.
It will need some where it will get very hard, very crumbly, and it will just go to dust. It actually is much softer when it rains.
It also pollinates, right?
Yeah, when it wants to reproduce, it will let off spores into the air, which will then find other spores and create reindeer lichen. Reindeer lichen is a mix between a moss and a fungus. It's over time they mutated into what we now call lichen.
Because it has certain properties that are like moss and other certain properties that are like fungus. Like generally how it breaks down is more like a fungus, and how it just grows is more like a moss.
What was the most exciting part of this project?
I would say that night that we went out late.
Oh yes, it was absolutely amazing weather. It was crystal clear. The moon was bright. We didn't even need flashlights after a certain point. Once it got dark enough that the moon actually, you could see the light. We didn't need flashlights anymore, and we just walked around and carrying the portable barometers. And there weren't very many insects or any at all. And there weren't any of the yeah, but we saw some bats flying around, and it was just really great.
We did the Cadance River Preserve Carnivores and Herbivores project. The purpose of this was to determine what sort of carnivores and herbivores attended the sites that were baited and at what times they went there. Our results were that we got a lot of deer at the herbivore site, several fishers, a fox, and someone's golden retriever at our carnivore site. And it was a pretty successful experiment. If our cameras had been set up better, it would have worked better. But other than that, it went well.
So at the vernal pool, we found these big bugs called giant water bugs, and she showed us two of them in these cases first, and we pretty much thought we were never going to find one. But then we did, and it was jumping around in the net, and so I like screamed bloody murder and ran off the dock. They're like this big and they bite, and every time I found one, I pretty much screamed off and ran. But towards the end, I got used to them, and they weren't as scary.
Actually picked them up in a spoon.
Yeah, I did. I picked it up in a spoon, which was a big step because that net has a pretty long handle.
All right, so we were the river invertebrate study, and we went out into the cat ants and we caught some. We used flat bottom nets and we scraped at the vegetation to get the invertebrates. And then most of the invertebrates are very small baby forms, yeah, like larva bugs. So then we counted and put them in charts, and then yeah, so we drew conclusions about how water temperature and depth affected different invertebrates.
And when things got colder, most everything started dying off, and
yeah, depth did not do wonders.
Depth did not do anything for it, but as soon as it got cold, things started dying in truckloads, and it was a good time.
We had 50 trees in our winter thinking plot, and we measured all of them. And we put in the data with the data from the last 4 years into a simulator, and it showed this is the plot as it is today, and then this is what it's going to look like in 100 years. And we also had 4 smaller plots outside of that are shown right there, and we measured those for ground covering, like the shrubs, the flowers, ferns, mosses, and we had 4 of them around the main plot. That graph shows the average tree DBH, which is the diameter at about 4 feet high, and the other graph shows the average tree heights by species.
Anything else mirror that on in the insect world?
The giant water bugs and the water scorpions went up as we kept visiting, but we actually figured out better ways to get them with the net, so we're not sure if it had more to do with the temperature or our strategy of getting them.
Did you stay right on the dock for your sampling?
Yes.
Like, did you use all 3 sides of it? And did you have a certain sweeping motion? Did you?
Yeah, we got the bottom of it sometimes. Sometimes we scooped up the grass because, like I said, different times we got different things. So the bottom we get more giant water bugs, and the grass we get more water scorpions. Just where they like to hang out.